Good morning, good afternoon, good evening, depending on when you listen to this podcast or watch it. What I told you about last week were the main processing steps of tRNAs and how they get made. And I finished by talking about the amino acid tRNA synthetases. And after our longer break, I will now continue by talking about the main role that you are familiar of tRNAs in decoding. And then in the second half, and this is not going to be required for the exam, I'll speak about what we've done in the field of tRNA modifications and quality control. What I put here in this presentation is something which may be helpful for you. So I have two examples of previous exams. This one I've already mentioned. So I asked for the full names of 5 nucleotides that you can actually find in tRNA. And a possible answer, and a very simple answer, would have been adenosine, uridine, guanosine, and cytidine, plus 1 pseudouridine or dihydrouridine we have mentioned a couple of times. But clearly adenine, uracil, guanine, cytosine, or pseudouracil would not be correct answers because these are the names of the nucleobases and not of the nucleotides. A second example of what I've asked in the past is the following. Here I'm asking for processes during tRNA biogenesis, but it could also be, in this case, translation or charging. Where do these things happen? Cytoplasm, nucleus, mitochondria. Here I gave away half a point per process, and when it was put in the right position. And you got 1 point for the right process and the enzyme. However, no points when the enzyme was mentioned alone. So you can see that you could probably, in any of those steps, you could pretty much almost gain 5 points. So theoretically, I haven't recounted this, but you would probably be able to get at least 10 or more than 10 points in this exam question. So in principle, this should be relatively simple. Now, let me jump a couple of slides back where we started. What I've told you is that you have a nucleophilic attack, which will lead to the charging of the tRNA. And this can either be in the position 3 of the sugar in the class 2 enzymes, and a similar reaction can also target position 2 in the class 1 amino acid synthetases. However, once the tRNA is at the ribosomes, it has to actually work always the same way. So all the tRNAs that have been charged in position 2 need to move to position 3 through a transesterification. So once this is all done, all tRNAs are charged similarly on the 3' OH. Now, the beauty of the synthetases is really that they are highly efficient and highly precise, because if they wouldn't be, then even the decoding wouldn't really matter if the wrong amino acid is put on the tRNA. So when you calculate the error rate of what you would expect in terms of thermodynamics, people end up with an error every 1,000 every 1,000 tRNAs or amino acids charged on a tRNAs. However, the error rate is much lower. It's actually by a factor of 10 lower. So 1 mistake in 10,000 charging reactions. And the question is, how does this work? So this works by the so-called double sieve strategy. And I highlighted this now slightly differently to show this actually to you. So this works because the synthetase has 2 sites, enzymatic sites. The first one is the active site that amino acylates the tRNA. And then you have not in every synthetase, but in many, you have an editing site. So this second site will basically correct mistakes that the enzyme has done before. And the way it works is, and that's shown here in this schematic, that the bulky amino acids, like in this case tryptophan and phenylalanine, they simply don't fit into the active site of the synthetase. However, smaller amino acids will all fit in. So practically, the synthetase will charge the leucine tRNA not only with leucine, but it will also accept isoleucine and valine. So which would lead to a mischarged tRNA. So while the first C structurally deflects bulky amino acids, it basically lets through also amino acids that do not work and are the wrong ones. And then in the second step, you again have an editing reactions of the editing site, which does the same thing, that it deflects certain amino acids which don't really fit. So in this case, the editing site will not recognize leucine, which is actually the correct amino acid, and will basically not react with it. However, isoleucine and valine, it will react with and then hydrolyze it. So the tRNA again is uncharged. And that means by first deflecting the bulky amino acids and then hydrolyzing the wrong but smaller amino acids, the leucine synthetase will actually only charge leucine onto a normal tRNA or the leucine tRNA. So this is kind of like, yeah, like what I say, a double check. But let's move to what tRNAs generally do, namely decoding. You all know this scheme, so but I would still mention again. So you have the three positions of a ribosome: the A site, the P site, and the E site. The A site is where most of the reading happens. So here the tRNA is brought by EEF1A, for example, in eukaryotes. That brings the tRNA to the anticodon. In the P site, that's where the tRNA sits with the amino acid chain attached to it, and the E site, that's basically where the empty tRNA is released. Now, this of course looks a little bit more natural and complicated in a real structure. So what happens is that there is a mini helix between the anticodon and the codon in the A and the P sites. So that's where really this codon-anticodon interaction matters. And you can see this here for the A site tRNA and for the P site tRNA. And then this gets lost at the end where the E site is, because you kind of see this kink of mRNA. So the mRNA doesn't continue, but it kind of moves down, making a kink, which then kind of releases this anticodon-codon interaction. And that's why the tRNA can be kicked out at the E site. Now, the way the selection works, again, through several steps, and they are all kind of kinetic reactions. So the tRNA bound, now we speak about the bacterial example where the elongation factor EFTU binds to the tRNA in its GTP bound form, and then basically samples the open A site. And if the tRNA seems to be wrong or clearly wrong, this is a situation where the tRNA bound to EFTU without GTP hydrolysis can simply be kicked out again. However, during the sampling process, and this is something that I'm showing you here, the tRNA really needs to go through a significant change in terms of its conformation. So you have now here in this structure the mRNA, and you can see that this is basically where the codon is located. However, the tRNA goes in slightly not matching it. So in order to really check and to really perform codon-anticodon pairing, it needs to reorient. And this is actually, on the one hand side, you can see this here, but in more detail zoomed in, you can see this here. So the tRNA really needs to be displaced by 15 angstroms and on the other hand rotate in order to really be able to match